Hi students, we're going to be talking about Chapter 17 or the digestive system, and you'll find this on two different videos. So this part is going to be the first part, part one of the digestive system, and it'll take us up until, up until the liver, and then part two is the end, starts from the end of the liver all the way to the end of the chapter. So here it goes. Okay, so a little intro on what the digestive system is. It's basically made out of a tube, and that tube opens out to the outside world, whether through the oral cavity or through the anal cavity. And the inside of the digestive tube is really considered part of the outside world. So it's affected by what's going out, by what's going on the outside. So, whatever we eat and kind of put into that digestive tract will pass through that whole tract. It really is not affected by what's going on in the internal environment. Although the internal environment, the changes in pH or the salt and water balance and all of that, can kind of affect what's going on surrounding the tube, but whatever is happening inside of the tube is not really related to those. What we have to do is that whatever we take in, all of the food materials that we ingest, as it passes through that tract, has to be broken down into its building blocks. And these building blocks could be those monosaccharides or sugars, could be amino acids if we're talking about eating protein, could be nucleotides, could be fatty acid and glycerol. With fatty acid and glycerol, both of those are the building blocks of fat. Okay, and we absorb those building blocks. Okay, we cannot absorb, say, a polysaccharide. Okay, polysaccharide, which is starch or sugar, we would have to break that down into its monomers of monosaccharides, and those building blocks, those monosaccharides, are what we can absorb. Bigger molecules like a disaccharide or a polysaccharide, those are too big and they cannot be absorbed into the digestive system, or they can't be absorbed through the intestinal cells. Okay, once we absorb those building blocks, the body can take them and use those to build bigger molecules all over again. So we could take those monosaccharides that we absorbed and build them into macromolecules. We can make now a polysaccharide like glycogen and store it and so on. We could take those building blocks of amino acids and build a protein. So a little overview. Here, that first off, we have to understand the difference between mechanical and chemical digestion. So digestion basically means that we're breaking down the food into smaller particles. And again, we break it down into mechanical digestion, which is basically chewing. Okay, so when we take a bite of that food and we start breaking it down into smaller pieces, that is what mechanical digestion means. It has nothing to do with the chemical composition of food. So we did not break down those bigger molecules into anything. We just broke down those bigger pieces into smaller pieces. And this is all on a macroscopic level. These are changes that you can see with the naked eye. Chemical digestion, though, this is when the enzymes and the digestive fluid, kicks in, and we break down those bigger chemicals into smaller chemicals, okay? And these are microscopic changes. So what is the function of the digestive system? It does not only include digestion and absorption, but it includes everything. So it includes, you want to start all the way from the top. So starting from ingestion or eating, and there's chewing or mastication, propulsion, which means pretty much pushing down what we will call chyme, which was the ingested food that we just ate, and mixing it up with all of the different fluids and enzymes and so on, and then digesting that, absorbing what we need, and then at the end, defecation of the waste products. Digestive system is made out of the alimentary canal, and that is the canal that again starts with the oral cavity all the way through until the external up until the anal cavity. And we also have the accessory organs. So we'll take a look here at the differences between them. So if we eat food and that food or bolus or chyme passes, is going to pass through through that tube. We're going to call that the alimentary canal. So the alimentary canal, again, starts in the oral cavity, made out of the pharynx, esophagus, stomach, small intestine, large intestine, and then again through the anus. So alimentary canal is the organs, or that they're pretty much kind of different tubes, pipes, where the food is going to pass through. Anything that helps with the process of digestion, but food does not pass through it, that is known as an accessory organ. So for example, salivary glands, those make saliva, but our food does not pass through these glands. The liver, the same thing, gallbladder, pancreas, which you can kind of see it behind the stomach. These are all considered accessory organs because, again, food doesn't pass through them, but they definitely help in the process of digestion. So what are the general characteristics of your alimentary canal? It is a muscular tube, so it's made out of smooth muscle fibers, obviously involuntary. We have no control over the passage of food throughout the whole alimentary canal. It's about 8 meters long, and it starts all the way up here, up in the head and neck, okay, and then passes through the thoracic cavity. So you have the esophagus, for example, that passes through the thorax. And then when it perforates the diaphragm, now we're into the abdominal cavity, and then, as you kind of go through the canal, parts of it, the lower parts are going to be in the pelvic cavity as well. So let's talk about the layers of the alimentary canal. Okay, so if we take one of those tubes and we make a section in it, we will find that they are made out of 4 different layers. These layers are consistent throughout the whole alimentary canal. So starting at the esophagus all the way down to the anus, you will find we have these 4 layers. From innermost layer to the outermost layer would be the mucosa is the first one, and that mucosa is made out of epithelium. And that layer, you'll find that it obviously is going to have it's a mucous membrane, so it produces mucus. It is also there for protection. It is going to secrete different things or absorb different things. So depending on where you are in the alimentary canal. Underneath the mucosa, you're going to find the submucosa. Sub means underneath, mucosa is underneath the mucosa. The submucosa has connective tissue. You'll find in there blood vessels. You'll find in there some nerves and lymphatic tissue and so on. Underneath that, the third layer is your muscularis. Muscularis is made out of smooth muscles, and these smooth muscles are responsible for pushing the food particles through the system. The serosa, that's the outermost layer. That's also made out of epithelium. It's connective tissue, and it makes a serous fluid, so this makes more of a watery kind of fluid, and it is there for lubrication and protection. So take a look at this image right here. This is a loop of the small intestine, and you'll see a section inside, and kind of take a peek of what's going on inside of the small intestine. That layer that you see here, that is the mucosal layer. Okay, so the mucosal layer is the innermost layer. And I'm not going to get kind of into the detail of what all of these finger-like projections are at this point. We'll talk about it when we get to the small intestine, but we're just going to use this kind of to take a look at the 4 different layers. So again, your innermost layer is the mucosa, and that has tons of mucus glands, those goblet cells that make mucus. So if you remember, goblet cells anywhere in the body are mucus factories. Then underneath the mucosa, there's the submucosa, and you can see the blood vessels. You can see the lymphatics. Those are the green structures. Underneath that, you have the muscularis. And the muscularis are smooth muscle fibers made out of inner circular and outer longitudinal muscles. Those, again, are going to help with the propulsion or the pushing of the food particles throughout the system. And then the outermost layer, that's that white layer at the very bottom, that is the serosal layer. And this is, again, made out of epithelium. It makes a watery fluid. And that watery fluid kind of decreases the friction between all of these intestinal loops. So So let's talk a little bit about the movements of the tube. Okay, so when we're talking about the tube, again, we're talking about the whole alimentary tract. So there are two different kinds of movements. One of them is a mixing movement, and the other is a propelling movement. So the mixing movement means that the food is not going to be moved around. Okay, it's in one area, and we're just mixing that food with different things. So for example, the example that you can see here is that the food, which we don't call it food anymore. Okay, so when we're swallowing it in the mouth, we call it bolus. And so we're going to swallow that bolus. When it reaches the stomach and it starts mixing with the gastric juice, that is now called chyme, C-H-Y-M-E. So, and we've got here the sphincters. This one is known as the lower esophageal sphincter, and this is known as the pyloric sphincter. Those are going to shut down. Both of them are contracted. So we've blocked the entrance and we blocked the exit, and the stomach is going to make its gastric juice. And in order to mix the gastric juice with the chyme, there is going to be these little segmentations that will help in that mixing process. Okay, so these are rhythmic contractions that happen in the smooth muscle fibers of the wall of the stomach. It also happens in the small intestine. So you can see here, there are contractions on either side, so the chyme isn't going to move anywhere. It's just being mixed with the digestive enzymes. That is, again, is a mixing movement. For example, segmentation. And so these segments right here, this process is known as segmentation. If you compare that to the propelling movement, that is going to be moving chyme in one direction. That is done by a wavy contraction, and that scientifically is known as peristaltic movement or peristalsis, where you have contraction on one side and relaxation on the other side, and that is going to push that chyme down the tract. Again, peristaltic waves happen in one direction, so chyme is going to be moving in this direction, but not in the opposite direction. Now, in order for all of this movement to happen, and the secretions that we haven't talked about yet, there has to be innervation to that tube. Okay, so there is a sympathetic innervation and a parasympathetic innervation. Know that the digestive system is something that all of this kind of happens automatically. Okay, so it does not have somatic nervous system control. It is under the control of the autonomic nervous system. Remember that parasympathetic is your resting and digesting part. So that is going to increase the secretion and increase the motility of the digestive system, while the sympathetic nervous system is more of your flight situation. So that when sympathetic impulses reach the digestive system, that's going to inhibit digestion. Okay, so it's going to inhibit both secretion and motility. And there are two different nerve plexuses in the alimentary canal. So when these parasympathetic or sympathetic nerves reach the parts of the digestive system, there are two different nerve plexuses. A plexus of nerves is basically kind of like a braid of nerves. Okay, so it's a whole network of nerves. There's one in the submucosa, and that's known as the submucosal plexus, and that controls secretions. So it's nice to know that all of the S's together. Okay, so the submucosal plexus controls secretions, and the other plexus of nerves is found in the muscularis level, and that is known as the myenteric plexus, and that controls motility. So if you take a look at the picture right here, you can see where those two plexuses would be. The submucosal plexus would be in the submucosal layer, and it controls secretions, while the myenteric plexus would be in the muscularis layer, and that controls motility. Now we'll start at the very top, the very beginning of the digestive system, and that is the oral cavity. And that's the very first part of the canal. This is where ingestion of food happens, and mechanical digestion starts. Mechanical digestion, as I said, means chewing the food, which scientifically means mastication. So you want to be familiar with these different terms like ingestion, which means eating, mastication, which means chewing, and you're also mixing all of that food now with the saliva. Okay, but the mouth has other functions, so it is also an organ of speech. Okay, it also has the receptors for taste, so it's a sensory organ. The oral cavity has the roof of the oral cavity as the hard palate, and then followed by the soft palate all the way in the back. The lateral walls are made out of the cheeks. And then we also have on the outer borders, there are the lips, and there's a vestibule. The vestibule is that little tiny space between the lip and between the gums. So for cheeks and lips, the cheeks are the lateral walls of the oral cavity. The cheeks are made out of muscles, okay? Those are skeletal muscles, so they have the muscles of facial expression and muscles of mastication. These are muscles that you can control, so those are skeletal muscle fibers. The inner lining of the cheek is a stratified squamous epithelium. So if you remember, stratified squamous was also the lining for skin. So the lining of the skin on the outside and the lining of the cheeks on the inside are basically the same kind. And that is due to the wear and tear that happens inside of the oral cavity. Now the lips, those have receptors for temperature and texture, and those are the boundaries between the skin on the outside and the mucous membrane that's found inside of the oral cavity. Now for the tongue. The tongue is also skeletal muscle, okay? It's a muscular organ, and it is found if the mouth is closed, it'll be on the floor of the cavity. If you flip the tongue upwards, it is connected to the floor of the oral cavity by this little part of mucous membrane that is known as the lingual frenulum. Okay, you'll find on the top of the tongue, you can find these little papillae, and that's where the taste buds are. At the very back of the tongue, and this is something that you really cannot even see in the mirror, okay? So I'll show you on the next slide that the tongue really extends all the way to the oropharynx, which is all the way to the back of the throat. So the lingual tonsils is not something that you can see if you're trying to take a look at it in a mirror. The lingual tonsils are a group of lymphatic tissue, and it tries to trap any germs that are getting in, trying to enter into our body with our food. So as you can see here, this right here is the tongue, and again, the lingual tonsils are all the way in the back, so something again that'll be very hard to see, trying to see it on your own. Now for the palate, and the palate is the roof of the oral cavity. The anterior part of the palate is made out of hard bone, and that's known as the hard palate, while the posterior part of the palate is made out of soft tissue, and that's known as the soft palate. The tip of the soft palate is known as the uvula, and whenever you ask the patient to open up and say, ah, you're looking at the movement of that uvula. On either sides of the uvula, there are palatine tonsils. We can only see, obviously, the ones that are on the right side in this section, but there are also palatine tonsils on the opposite side. And there are other tonsils that are found all the way up here in the nasopharynx, and those are known as the pharyngeal tonsils or adenoids. Again, all of these tonsils are just scattered, lymphatic tissue surrounded by connective tissue, again, trying to trap germs entering the body, whether it's through the oral cavity or trying to enter through the nasal cavity. When we say tonsillectomy, so by default, we really mean the removal of the palatine tonsils. Now for teeth, teeth are by far the hardest structure in the body. They are not bone, okay? They are much harder than bone tissue. We have primary teeth. Those are known as the deciduous teeth, and those are 20. So 10 on the top and 10 on the bottom. And we also have the permanent or secondary teeth. Those are 32. So 16 on the top or on the upper jaw and 16 on the lower jaw. Here you could see those little tiny deciduous teeth, and these are the secondary teeth. And as they grow, they would push those primary teeth out and then take their places. So this right here, we're going to kind of go over the different names of the teeth. These are your secondary teeth. Okay, so we'll be talking about, 32, 16 upper jaw, 16 on the lower jaw. And if you imagine a straight line going through the middle, you'll have the two that are closest to the midline. Those are known as the incisors. So there's a medial incisor and a lateral incisor. And then there's a canine or bicuspid tooth, and that is the one that kind of has a sharp tip. And then there are two premolars or bicuspids. And then we have three molars. Okay, and you can see that pattern would be the same on the opposite side, and it repeats on the lower jaw as well. While we have this open here, this right here is the uvula, and these two are the palatine tonsils. Now we'll take a look at a section of the tooth. Remember when we were talking about joints, we said that the tooth and socket, or the tooth is a gymphosis kind of joint. If you don't remember that, you can go back and take a look at that. We were talking about basically having some tooth that is, is found in a socket. Okay, that socket is found in the jaw, whether it's the upper or the lower jaw. And this little socket is known as your alveolar process. The upper part of the tooth, or the part of the tooth that we can see, is known as the crown, and that's made out of the white enamel. This is the hardest part of the tooth. And then underneath the enamel there is dentin. In the middle of the tooth there is a cavity, and that's known as the pulp. And in the pulp you can find the arteries and veins, the blood supply, and you can also see these yellow nerves. Okay, that would be the nerve supply to that tooth. Now the part of the tooth that we do not see, okay, that's hidden in the gingiva or our gums and inside of the bone, that is the root. And down here in the mandible, this is where the blood supply and the nerve supply is coming from, and it will enter into the tooth through a root canal. Now what keeps that tooth in place would be these periodontal ligaments. So these very, very tiny ligaments that, again, keep the tooth in place. It's not until, as we age, these odontal ligaments are going to get looser, and then sometimes these teeth would fall. Now for dental caries or dental cavities, those would be cavities in the enamel of the tooth, and it's formed because there would be, as we eat throughout the day, some food is going to stick to the surfaces of the teeth, especially in the areas between the tooth and the gingiva. So usually in this area, and that's why we're always instructed to floss in order to get to these hard to reach areas. And when food particles remain there, especially if they remain for a long period of time, like overnight, okay, bacteria is going to ferment the food and metabolize it into acidic products, and these acidic products are able to destroy the enamel and destroy the dentin. So the recommendations of the ADA, which are the American Dental Association, would be to brush and floss, at least twice a day, dental exams and cleanings at least twice a year, fluoride treatments as needed, and sealants if needed. Now moving on to the salivary glands, those are going to make saliva, pretty straightforward. Okay, saliva helps in the moistening of food as we eat. It binds it together and makes the food into a bolus. Okay, but that's not the only function of saliva. Okay, saliva also has enzymes, and we're going to start the digestion of carbs in the oral cavity. The saliva has an enzyme known as salivary amylase. And salivary amylase is the very first enzyme. Again, it will start the breakdown of those polysaccharides into smaller disaccharides. So there's bigger molecules of sugar into smaller molecules made out of only two sugar building blocks. But obviously we don't keep food in the mouth for a long time. So we'll meet other amylase enzymes to help. Okay, there's also bicarbonate ions, and that is to neutralize the effect of remember the bacteria in the oral cavity makes lots of acid, so you want to neutralize that by bicarbonate ions found in the saliva. And we've got tons of salivary glands. Okay, the three big ones are the parotid, those are by the ears, the submandibular gland, that's underneath the mandible, and the sublingual glands, and those would be underneath the tongue. Okay, there are pairs, okay, so there's a right and left of each of these. But we have also minor glands, salivary glands, that are scattered throughout the mucous membrane of the oral cavity. So they're found on the inner side of the inner side of the cheeks, on the palate, on the surface of the tongue, and so on. And those are to keep the lining of the mouth moist. Now, all of these glands make saliva, which is true, but we have two different kinds of saliva. So we have two different kinds of cells to make these two different kinds of saliva. We either have these serous cells, and serous cells are going to make a very watery kind of saliva that has lots, is very rich in digestive enzyme that we name salivary amylase, and that's going to split your carbs, whether it's a starch or glycogen, into disaccharides. But remember, again, it doesn't finalize it, okay? So we have, we don't keep the food in our oral cavity long enough in order to digest all of the starch and all of the glycogen into disaccharides, but it starts the process. We also have mucus cells, and those mucus cells are going to make mucus, and that helps to kind of bind the particles and lubricate, especially when we're swallowing. So the three major pairs of the salivary glands, first one is the parotid, and that, as I said, you'll find those right in front of the ear. Those are the biggest of the salivary glands. These make a very serous secretion. Okay, so this is more of the watery saliva rich in the digestive enzymes, amylase. And so for each one of these, you want to know, where is the gland? Okay, what kind of saliva does it make? Is it more serous? Is it more mucus? Or is it a combination of both? And where do the glands open? So each of these glands have ducts to take the saliva that was made and dump it into the oral cavity. So where do these ducts open? So for the parotid duct, and I'm going to kind of go to the picture, this right here is the parotid gland found in front of the ear, and that would be the parotid duct. It crosses right on top or superficial to the masseter muscle, and then it perforates this muscle called the buccinator muscle, and it opens up across the second upper molar tooth. And remember, again, this produces more of the serous saliva that is rich in amylase. Okay, the next one, which is the sublingual gland, that makes a very mucus kind of secretion, and those have many separate ducts. So you can take a look at this right here. This is the sublingual gland. That's the tongue. This is a gland underneath it. Remember, we have two of them, so there's a right and left of these. And then you can see that we have multiple smaller ducts that are going to open up into the floor of the oral cavity. Now, the third is the submandibular gland, and this is found underneath the mandible. Again, there's a right and left of these. This makes a mixture of saliva, so it has both kind of like, half serous, half mucus. And you can see the submandibular duct that opens as well in the floor of the oral cavity on either sides of the lingual frenulum. So is there a picture? Maybe the other side was the picture. Bear with me for 1 second so we can get to that picture really quick. Okay, so on either sides of this lingual frenulum, there will be two openings, and you can actually see this in the mirror if you take a close look, or if you, inspect the oral cavity of somebody. You could see these little two openings on either sides of the lingual frenulum. Those would be the openings of the submandibular salivary gland. Now that we're done with the oral cavity with the accessory glands that we just talked about, the food is going to move on, or now we call it bolus, move on to the pharynx, which is the back of the throat, and then down the esophagus to reach the stomach. So let's talk about the pharynx. Pharynx is an organ that is part of the digestive system, and it's also part of the respiratory system. So it's a pathway for both food and air. Okay, the pharynx is made out of 3 different parts, and it's made out of these 3 strong muscle fibers. These are those are known as the constrictor fibers. So there are superior, middle, and inferior constrictor muscles. These muscles are very strong muscles, and they kind of act like the, if you've ever seen a snake eating something, okay, so you know how boa constrictor, for example, this is where the name comes from. If a snake when they start eating something, you could see kind of like these wavy contractions in order for it to swallow their prey. That's the same thing what our pharynx or pharyngeal muscles do. Okay, so these superior, middle, and inferior constrictor muscles do the same thing. The pharynx, if you remove the muscles and you take a look at the tube that's inside of those or underneath those muscles, you'll find that we have a nasopharynx. So this part of the pharynx opens up into the nasal cavity. This part of the pharynx that opens up into the oral cavity is known as the oropharynx. And then this part that opens up into the larynx, so this right here is the larynx, or where the vocal cords are, and this part would be known as the laryngopharynx. It'll be continuous down here with the esophagus, but you can see here that it always also opens up into the trachea, which is the windpipe. So as we're swallowing, we have this little dilemma here that food might actually get into the trachea instead of going down the esophagus. So there is a reflex for swallowing in order to shut down, to make sure that the trachea is closed and the nasal cavity is closed as well, so that food doesn't go upwards into the nasal cavity. So we'll talk about the swallowing mechanism. Okay, we divide it into 3 different stages. The first stage, and that's the voluntary stage, and that's when you're chewing and mixing the food with saliva and you form the bolus. Okay. Second stage, and this is, when we push the food all the way towards the back by using the tongue, we'll push that bolus towards the pharynx, and when it touches the oropharynx, this is going to trigger the whole swallowing reflex. So we'll take a look at how we overcome, what is going on. So remember, we said we have to close the nasal cavity. So you'll find this uvula will flip upwards. And then this epiglottis, this is a cartilage known as the epiglottis. This epiglottis is going to bend downwards and shut down the trachea. You can see here better pictures of this. There you go. Okay, so this right here is the tongue pushing that bolus of food towards the back. And this again is going to initiate or start or trigger the swallowing reflex. Once we do that, you find the soft palate here going upwards and closing the nasal cavity. You could find here this epiglottis that used to be flat or straight. It is going to bend and close the trachea so that the bolus of food is now going to be pushed. It has really no other way to go except towards the esophagus. So that would be your second stage. Now, the third stage would be the peristaltic movement that pushes the peristaltic movement down the esophagus into the stomach. And the only part of the swallowing reflex, or the only part of the whole swallowing process that is voluntary, is really the first phase, which is the chewing portion. Now that we're into the esophagus, let's take a look at what it is. So the esophagus is a muscular passage from the pharynx into the stomach. It passes through the thoracic cavity, and then it is found behind the trachea, and it has to perforate the diaphragm in order to reach the abdomen, and the opening in the diaphragm that allows for the passage of the esophagus is known as the esophageal hiatus. There's a sphincter at the very bottom as it opens up, or as it that would have to open, the sphincter would have to relax in order for that bolus to enter the stomach, and that is known as the lower esophageal or the cardiac sphincter. It has two names, and this regulates the passage of food into the stomach. The esophagus has absolutely no role in digestion or in absorption. So the esophagus does not make any digestive enzymes. It does not absorb anything. It is just a pipe for the bolus to reach, to go from the pharynx all the way to the stomach. Now that we reach the stomach, we want to talk a little bit about the structure and then what is the function of the stomach. So the stomach is again, has those four layers that we talked about, the mucosa, submucosa, muscularis, and serosa. The muscularis, though, instead of only having the inner circular and outer longitudinal layer, there's a third layer of muscle, and that is the innermost layers, and those are made out of oblique fibers. So the stomach, there's a lot of movement going on in the stomach. Remember, we had the segmentation movement, and we have to mix whatever we ate, that bolus, with lots and lots of digestive fluids. So stomach, you need that kind of extra layer of smooth muscle fiber. The stomach is J-shaped. Okay, it's found right underneath the diaphragm on the upper left quadrant of the abdomen. If you open up the stomach, it's made out of lots of folds of mucosa. Those folds are known as rugae, and it allows for the stomach to distend as we eat. So what is the function of the stomach? Well, it receives food from the esophagus. Okay, it makes digestive enzymes and lots of other things like hydrochloric acid, and we collectively call that as gastric juice. It starts the process of protein digestion. There's very limited absorption in the stomach. We'll talk about what are the things that our body can absorb from the stomach. And then after we mix the bolus with all of the gastric juice, all of that is going to move on to the small intestine. Okay, so you see here that I have this underlined. So we started the digestion of carbs in the oral cavity through salivary amylase. We are going to start protein digestion, though. Nothing happens to protein until it reaches the stomach. So the very first step in protein digestion happens in the stomach. These are the different parts of the stomach. So we'll kind of go over the smooth muscle fibers. You can see here the outer longitudinal, the inner circular, and then you have these innermost oblique layers. If you open up the stomach, you can find that this right here is the entrance. That is the lower esophageal or the cardiac sphincter. It is usually contracted, meaning that it's usually shut. When we swallow, part of the swallowing reflex would be to relax that sphincter so that bolus can enter into the stomach. There's another sphincter, and that is the exit of the stomach. That's known as the pyloric sphincter. That is going to remain shut as well until everything is mixed up with the gastric juice and it's ready to move on to the small intestine. The bigger curve right here, this curve is known as the greater curvature of the stomach. This right here is known as the lesser curvature. And then we divide the stomach into this upper dome part that's known as the fundus. The majority of the stomach is made out of this body. And then the lower part of the stomach is known as the antrum. And then we also have that pyloric canal, that kind of looks like a funnel that ends in this pyloric sphincter. You can also see those waves or those rugae that are found inside of the stomach, and so that when we eat, the stomach can distend and kind of accommodate the amount of food that we ate. The lining of the stomach, exact same thing. So we have the mucosa, submucosa, muscularis, and the serosa. Just remember a couple of differences, that the muscularis here has a third layer. So why are we showing this? So that we can take a look at this gastric gland. Okay, so all of these waves right here, the dips are known as a gastric pit, and this is where the gastric juice or the secretions are going to be coming out of. So you'll see lots and lots of mucus cells scattered throughout the epithelium. Those are going to make mucus. You can also see parietal cells, and at the very bottom you can also see chief cells. So these chief cells are going to make enzymes, the protein digesting enzymes, And these digesting enzymes are going to rise through these gastric pits in order to reach the cavity of the stomach. These parietal cells make hydrochloric acid that are needed to activate these enzymes. And it also makes a chemical known as intrinsic factor. Intrinsic factor is important in the absorption of vitamin B12. So if somebody has these parietal cells destroyed or had a gastrectomy, which is removal of the stomach, and they no longer have enough of these parietal cells, they're no longer making enough intrinsic factor, then they are no longer absorbing vitamin B12. That leads to a kind of anemia that we talked about when we talked about the blood, chapter 14, that we named megaloblastic anemia or pernicious anemia. So again, we'll try to introduce now the different parts of of gastric juice. So we have the chief cells, and those are the cells that are all the way down here at the bottom of the gastric pits. Those make pepsinogen. So remember, whenever we end a word with gen, that means it's going to generate this. Okay, so pepsinogen is going to be activated into pepsin. This is an inactive enzyme that digests protein. You will find that throughout the digestive system, any enzyme that digests protein is secreted in an inactive form. Okay, and that is because we don't want them activated unless there's actually protein to digest. Remember, the stomach, our whole body is made out of protein, so unless there's food to digest, we're going to keep them in an inactive form. So again, pepsinogen is an inactive form of pepsin. It's made by these chief cells, and it is activated by hydrochloric acid. Okay, this is going to start the digestion of proteins. We are going to break down those polypeptides into smaller dipeptides or even just shorter polypeptide chains. It also makes another enzyme that's known as gastric lipase. So from the name, you can see here that it is going to start the digestion of lipids. And because this enzyme is made by the stomach, we're going to call it gastric lipase. You'll see that other lipases are made. So for example, you'll find a pancreatic lipase, and it's nicely telling you that is the lipase made in the pancreas. The one that the stomach makes is known as gastric lipase. It's also secreted by the chief cells, and it starts the splitting of those triglycerides into monoglyceride and 2 fatty acid chains. Hydrochloric acid, these are made by the parietal cells, okay, and that decreases the pH inside of the stomach to very, very low levels. So the pH in the stomach is about 3, which is a very acidic environment, but that is necessary in order to activate pepsinogen. Intrinsic factor is another factor made by the parietal cells, and as I mentioned before, that intrinsic factor is going to be taken all the way down into the intestine, and it helps in the absorption of vitamin B12. Without having intrinsic factor, we are unable to absorb vitamin B12, and that leads to a kind of anemia known as pernicious anemia. Last but not least, our mucus cells are going to make mucus, and that helps in the lubrication process, and it also helps in kind of protecting us from all of that hydrochloric acid that is being made. So how does the body regulate gastric secretions? Like, how do we know? So that you know, generally speaking, we are making gastric juice all the time, but at different rates. So how does the body know that, I'm about to eat, now I need to make more gastric juice, or I'm going to fall asleep, so I do not really, or, I'm about to, there's no food in my stomach. I can now, decrease the amount of gastric juice I'm making. So that is what we mean by regulation of gastric secretions, because it is a continuous process. So it is under the control of the nervous system and the endocrine system. So neural regulation, and I hope you guys at this point are able to guess, sympathetic stimulation would decrease gastric activity. So again, when you're in a fight, when you're nervous, under stress, okay, under sympathetic control, this is not the time to be digesting your food. This is when your gastric juice production is going to decrease. Under parasympathetic, though, this is when we are ready to digest, we are relaxed. That is going to stimulate gastric secretion. Hormonal secretion in the stomach, as the stomach, receives food, that is going to lead to the release of a hormone in the stomach known as gastrin. Gastrin increases gastric juice secretion. Okay, we've got two other hormones, though, somatostatin and CCK. Somatostatin and CCK, those are hormones that come out of the duodenum, which is the very first part of the small intestine, which technically means that there's no food in the stomach anymore. Food has moved on, left the stomach, and started to move into the small intestine. So the small intestine is going to make these two hormones to inhibit gastric secretion and inhibit gastric motility. So somatostatin with an S decreases gastric secretion, and CCK is going to decrease gastric motility. So you can see here kind of how these impulses work. So you have your parasympathetic fibers, those that are carried on the vagus nerve, which, if you guys remember, was your 10th cranial nerve. That is going to increase the production of gastric juice. So it could be very easily, you don't even have to eat. You can think about food. You can smell food. You could, see a picture of food, okay? So all of these different things that are happening in the head, okay, so this is known as the cephalic phase of regulation, where these impulses are going to stimulate the secretion of kind of preparing the stomach to receive food. Okay, once food gets into the stomach, that is going to release gastrin. And gastrin goes through the bloodstream, and then it goes back to the stomach and stimulates the production of more gastric juice. So to kind of understand how this gastrin works, okay, I'm just going to go back to a microscopic picture of the wall of the stomach. Gastrin is going to be produced, by these cells, and it does not go into the lumen of the stomach, okay? It goes to the bloodstream. So remember, hormones are kind of released into the bloodstream. It has to circulate in the bloodstream and then come back to the stomach in order to produce an effect. So that's what gastrin does. So gastrin made by the cells are going to circulate in the bloodstream and then go back to the stomach and stimulate gastric secretions. So these are, again, the three phases of gastric secretion. So cephalic phase, this is before you eat. So again, just smelling food, thinking about food. This is mediated through the parasympathetic or the vagus nerve, and that increases gastric secretion, prepares your stomach to receive food. Gastric phase means that you're actually now eating, okay, and that is is mediated by gastrin. Now this gastrin is going to stimulate your stomach to make even more gastric secretions. The intestinal phase means that the food has now moved on from the stomach, has moved into the small intestine. You no longer need gastric secretion or motility. So this is when the intestinal cells are going to make CCK and somatostatin. Somatostatin decreases gastric secretions, while CCK decreases gastric motility. CCK, by the way, stands for cholecystokinin, and we'll go over, more about this hormone in a bit. Okay, so gastric digestion and absorption. We already kind of just mentioned that there's very limited amount of absorption that happens in the stomach, but the stomach can absorb some salts and water. So you'll always find that when we talk about absorption of salt, water is going to follow by osmosis. We are able to absorb some lipid-soluble drugs, and alcohol is also absorbed from the stomach. Okay, we start the process of protein digestion through pepsinogen that is activated into pepsin. We also start the digestion of fat, very limited though, by gastric lipase. Mixing and emptying actions of the stomach, those are basically where the bolus of food reaches the stomach. we would have to shut down both sphincters, so contract the cardiac and the pyloric sphincter, do that segmentation, okay, where you're mixing now all of the gastric juice with the food, and we make it into kind of a paste, and we call that paste chyme. And then the pyloric sphincter is going to relax and start emptying the gastric contents into the first part of the small intestine that is known as the duodenum. These mixing and emptying actions are under control of your parasympathetic system as well. Okay, so you see here the parasympathetic, it actually starts here with the emptying. Okay, so the duodenum is going to start filling up with chyme. That's going to stretch the wall of the duodenum, so you're now stimulating those stretch receptors through a sensory neuron that is going to reach the CNS. Okay, now the CNS is going to tell the stomach to kind of slow down. You do no longer need to move a lot, so to decrease the peristalsis, and that whole reflex is known as the enterogastric reflex. A lot of the reflexes in the digestive system, if you look at the name, it'll tell you where it starts and where it ends. So the enterogastric reflex, it starts in the small intestine, starts in the duodenum, and ends in the gastric or in the stomach. So that stretch in the duodenum decreased the gastric motility. Now, a very common problem that you'll find out there would be heartburn. Okay, heartburn is a misnomer, meaning that the heart is not really on fire. Okay, it is just due to the acidity going up into the esophagus, going in the wrong direction, and that could lead to that sense of that kind of pain that is felt in the area. Okay, it usually occurs due to eating a lot of food too fast. And that's why individuals that do have heartburn, they are instructed to eat at a slower rate, take smaller bites, chew very carefully, and to take a long period of time to eat. So instead of eating, in 5 or 10 minutes to kind of slow down, not only is it healthier and it helps in digestion, it also gives your satiety center, which is your hunger center in the hypothalamus, it gives it time to realize that you have a full stomach, and it decreases the amount of food that we would eat per meal. It takes about 20 minutes for the hypothalamus to realize that your stomach is full. So the slower you eat, you're giving your hypothalamus more time to realize that that you're full and you no longer need to eat so much. Okay, if that doesn't occur, as we eat, the more we eat or the faster we eat, or sometimes it's just due to weakness in the lower esophageal sphincter, hydrochloric acid is going to regurge into the lower part of the esophagus, leading to inflammation, and that is known as esophagitis. The antacids can lead to some relief, but you would like to prevent it by eating smaller meals slowly. Do not eat and go to sleep, okay, to stay up right after eating, to try to avoid as much as possible of caffeine, nicotine, and alcohol. All of those increase the incidence of heartburn. Now, moving on to the pancreas. Pancreas doubles up as an endocrine gland and an exocrine gland. We talked about the endocrine portion of it, where it makes insulin and glucagon, and how it helps in the regulation of blood glucose. When we talked about chapter 13, this portion we are talking about the exocrine portion of it, which would be the making of these digestive enzymes. Okay, and that fluid is known as the pancreatic juice. This I kind of, borrowed this picture from another course where you could see the different parts of the different cells. So these cells right over here are kind of gathered together into their own island, and we call those the islets of Langerhans. Langerhans is the name of the scientist that discovered them, and he named the islet after himself. These cells make the insulin and glucagon and somatostatin. But this portion that has a duct, that's the exocrine part of the pancreas. we have these acinar cells. These are going to make the digestive enzymes, while these duct cells secrete bicarbonate. So you'll see here the pancreatic acinar cells. Those are going to release the pancreatic juice that have your enzymes. We also have the pancreatic ducts. Okay, I guess I could use the picture to describe that. So that bigger pancreatic duct then divides into a major pancreatic duct and a minor pancreatic duct. Both of them eventually open up into the duodenum through this minor duodenal papilla and a major duodenal papilla. A papilla means a bump. So on the inside of the duodenum, there are two bumps. One on the top, which is smaller, known as the minor duodenal papilla. The bump on the bottom is bigger, and that's known as the major duodenal papilla. You see here that there are two different ducts opening into this. We have the major pancreatic duct, and we also have this common bile duct, or just, we could also name it the bile duct. Both of them open up together into this major duodenal papilla. There's a sphincter there, so if you kind of take a closer look here, this is your pancreatic duct and your bile duct, and they both open up into the same opening. But you can see a sphincter. That sphincter is known as the hepatopancreatic sphincter. So hepato coming from liver, the bile portion. Pancreatic means the pancreatic portion. It's known as the hepatopancreatic sphincter or the sphincter of Oddi, O-D-D-I. So if you see both of these, we're talking about the exact same sphincter. The hepatopancreatic ampulla and the hepatopancreatic sphincter. So the ampulla is basically the opening into the papilla, and the sphincter are those smooth muscle fibers that we talked about. These smooth muscle fibers are normally contracted, and in order to dump the pancreatic juice and bile into the duodenum, these fibers have to relax. And that is the function of cholecystokinin, or CCK. Pancreatic juice is made out of these different things. Pancreas is really the organ that makes the most enzymes that help in the digestion of everything. So they digest carbs, fats, proteins, and even nucleic acids. So we have pancreatic amylase, and that would split starch and glycogen, or our carbs, into disaccharides. It'll break down pancreatic lipases into these triglycerides into monoglycerides and two fatty acid chains. All three of these help in the breakdown of protein. So there's trypsin, chymotrypsin, and carboxypeptidase. All of these are secreted in an inactive form. So trypsin is secreted as trypsinogen. Okay, and then it is activated by an enzyme found in the small intestine known as enterokinase into trypsin. Chymotrypsin is activated by trypsin, so it is released as chymotrypsinogen, and then it is activated by this enzyme. Carboxypeptidase is also released in an inactive form. I'm not even going to try to pronounce it, but it is also activated by trypsin. Okay, nucleases, that helps in the digestion of nucleic acids. And then there's lots and lots of bicarbonate ions to neutralize all of that acid that is coming out of the stomach. Okay, so it's a buffer for all of that stomach acid. Now, if we're secreting something, we always have to have a way to regulate it. How do we regulate the hormones, or how do we regulate all of that pancreatic juice? There are two hormones that come out of the duodenum that regulate it, and those are secretin and CCK. Secretin is going to stimulate the pancreas to make lots of bicarbonate. Okay, while CCK, or cholecystokinin, is going to, it does a couple of things. Okay, so it stimulates the contraction of the gallbladder, and it also helps in the relaxation of the sphincter of Oddi. If you break down the name cholecystokinin, cholecyst means gallbladder, kinin means to activate. So CCK activates the gallbladder by contracting it, It'll push all that bile in. Okay, now that the bile and the pancreatic juice is here, we would have to relax that sphincter. So the function of CCK is to contract the gallbladder and relax the sphincter of Oddi. Okay, remember that secretin and CCK, both of them are made by the duodenum. They are going to circulate in the bloodstream till they reach the pancreas, leading to the production of pancreatic juice. Secretin increases the production of bicarbs, while CCK is going to stimulate the production of those pancreatic digestive enzymes. Now moving to the largest organ of the body, internal organ, because the largest organ is skin, but the largest internal organ of the body is liver. It is found in the upper right quadrant of the abdomen, right underneath the diaphragm. It is a very dark reddish organ, and that color comes because of its very high blood supply. Okay. The liver has 4 lobes. Okay, so there is the right lobe, that's the biggest of the 4, and there's a left lobe. And then in order to see the 2 other lobes, you have to flip the liver over. So this is the inferior surface of the liver. You can see a caudate lobe. Caudate means tail, so this is towards the back where the inferior vena cava is. And there's also a quadrate lobe, and that is on the side of the gallbladder. Where different things enter or exit the liver, that is known as the hilum of the liver. So a hilum, H-I-L-U-M, of any organ is where things enter or exit out of that organ. If you break down, you kind of look at the microscopic structure of the liver, we break down the liver into these little hepatic lobules. These are the structural units of the liver. You need to have tons and tons of functioning hepatic lobules to have a healthy liver. Hepatic lobule is made by this central vein surrounded by all of these liver cells known as hepatocytes. In between the hepatocytes, we have these little lakes of blood, and those lakes of blood are known as sinusoids. So just want to take a look here. So this is a bigger picture. We can kind of look at that. And before we take a look at the different parts, we want to understand the blood supply of the liver. The liver gets 2 different blood supplies. It gets an artery with oxygenated blood. Okay, so this red artery right here full of oxygenated blood, just like any other organ, and that is known as the hepatic artery. It also gets everything that we absorb from the intestine, all of those goodies, the sugar, amino acids, and everything that we absorb from the intestine has to go to the liver first. And that is deoxygenated blood. Okay, that was blood that was already used by the intestine, bringing in all of the absorbed material, and that would be through the hepatic portal vein. You kind of want to think of the liver as the kitchen of the body. Okay, the liver is going to take all of these ingredients that we just absorbed and start cooking it up into different things. So again, your liver gets 2 blood supplies: oxygenated blood from the hepatic artery. Okay, this is a branch of the aorta, while it gets deoxygenated blood from the intestine that has all of the absorbed material, and this is known as your hepatic portal vein. Now, both of these are going to dump that blood, okay, into these sinusoids. So the spaces in between the hepatocytes is known as sinusoids, and sinusoids has a mixture of oxygenated and deoxygenated blood. All of that will be dumped in here. Now these hepatocytes can use the oxygen from here and all of the goodies that we just absorbed and make it into different things. So it can make either proteins using the amino acids, or maybe make glycogen using all the glucose. Whatever the body needs at that point, these hepatocytes are going to make. You can also see these little macrophages that are found in the sinusoids. They are known as Kupffer cells. So Kupffer cells are just fancier macrophages that are found in the sinusoids of the liver. Now that blood is going to be drained, okay, into the central vein, Now, all of that, now what is definitely deoxygenated blood is going to go into the inferior vena cava and then eventually into the heart. You can also see all of this green material. So that is bile. Bile is made by the hepatocytes. Bile is made by the liver. And you can see it here being dumped into these little bile ducts, and then they are going to be collected and stored into the gallbladder. Okay, so the major functions of the liver, okay, in all metabolism of everything. So remember again, the liver is considered your kitchen. Okay, so it is going to metabolize carbohydrates. It could take glucose and build it up into glycogen, or in cases, say, of fasting, it could take that glycogen that it stored and break it down into glucose. And now we could use glucose. This process of making glycogen is known as glycogenesis, while the process of breaking down glycogen is known as glycogenolysis. It can convert non-carbs into glucose, so it has the ability to take amino acids or fatty acids and change it into glucose. And that process is known as gluconeogenesis, or the generation of new glucose. Its role in lipid metabolism, that it could oxidize fatty acids. So instead of burning glucose for energy, it could burn fatty acids and make energy. It could make lipoproteins, so add lipid and attach it to a protein to make different lipoproteins. Or it can make phospholipids, it can make cholesterol. It all has just the ability to kind of, take different ingredients and cook them up and make other stuff. In protein metabolism, it could take those amino acids and break it down or use them to make plasma proteins. Okay, it could take amino acids and convert them into other kinds of amino acids. The liver stores different things. So it stores glycogen, which is basically glucose stored in the liver. Whenever we need it, it will break it down and release that glucose into the bloodstream. It is also a storage for vitamins A, D, B12, iron, and for even blood. Okay, so remember all those sinusoids. Those are considered kind of like blood reservoirs that we can tap into if needed. It has a role in blood filtering. So remember all of those Kupffer cells, those hidden macrophages in the sinusoids, those are going to filter out germs. And it also helps in filtering the damaged and old RBCs, okay, and removing them by phagocytosis. So remember we talked about the breakdown of old RBCs and how we break it down, hemoglobin into hemoglobin, and then we use that to make bilirubin and so on. All that process occurred in the, if you remember, we talked about it happening inside of the liver. The liver can also detoxify. So any drugs, any medications that we take we will detoxify them or metabolize them in the liver. That is why if somebody has liver problems or liver failure, you definitely have to readjust the dose and give them lower doses than normal. The secretion role that it has is that it can produce and secrete bile. Okay, so this is where I'm going to stop because the second portion that is already up there, I recorded that last semester. I think, if I remember correctly, I think it was a snow day, and I went ahead and recorded it for them. So I'm gonna, have you guys listen to that as well. Let me know if you have any questions.